Good morning, everyone. Welcome to Virtual Nutrition. It's been completely crazy. I know that we barely meet to begin with. Last class, we started a lab and we didn't get to finish that lab. Now we're not in school. So we will start where we left off with, section 4.2. But before we get into that, I do want to review again briefly what we learned from 4.1 and then also a little bit about enzymes. So just to refresh everybody's memory, we're on chapter 4, carbohydrates, sugar, starches, and fibers. In 4.1, we talked about the difference between monosaccharides, disaccharides, and polysaccharides. Now, just to bring back why we need carbohydrates. We need carbohydrates to fuel our body, mostly glucose. Glucose is going to fuel our brain. It's also going to fuel our muscles. It's going to provide energy for our body. Also, glycogen, another monosaccharide carbohydrate, which is going to fuel our muscles. And we also need fat, which we're going to talk about later in a different chapter. We did mention that people don't eat glucose and glycogen, right? But they eat foods that are rich in carbohydrates so our body can break it down to receive glucose and then also convert some of that to glycogen as well. Now, we talk about in 4.2, talking about these, enzymes in our body that are going to help us digest carbohydrates. These enzymes are helping the chemical reaction of hydrolysis. So what is hydrolysis? Hydrolysis is what's going to break a disaccharide into two, right? It's going to break it down using what? Using water. So here we have a disaccharide. What disaccharide do we have? Maltose in this picture. So what is maltose made of? What are the two monosaccharides that make up maltose? So here we have two molecules of glucose because that's what makes up maltose. And water is going to be used to break the bond. So the common atom that is bonding these two molecules together is this oxygen atom. So if we have hydrogen bonding to the oxygen and an OH bonding to this molecule, we will get two molecules of glucose. Why? Because that missing link, when they're in the maltose form, when two glucose are bonded together, they're bonded by one oxygen. When in reality, when they're by themselves as two glucose molecules, there's an OH bond here, two OH groups. So each one needs one OH group. How are we going to do that? By bonding a hydrogen with the oxygen, and then that being for one glucose molecule, and then an OH going to the other molecule. So using water to break the bond between a disaccharide. This, and you can see it right here, common during digestion, which is exactly what we talk about in 4.2. And then just briefly, a little review about starches. So starches are glucose molecules linked together. Okay, and that's important when we talk about digestion. But all of these, polysaccharides, these complex carbs, contain many molecules of glucose, and that would be glycogen, starches, and fibers. Many molecules of glucose linked together. Okay. So I know I gave you that note sheet to fill out while you're reading 4.2. I'm not sure how many of you left it at school, how many of you remember to do it, but what I did was I posted that on Google Classroom. So while I'm going through this lecture, questions, as you can see, are already popping up. And also while I'm going through this lecture, please, if you haven't already, use that Google Doc or use your physical copy. Fill out notes either during the lecture or after the lecture while you do your reading. But once you fill out those notes, if you do it on Google Classroom, great. If you do it on your physical copy, just make sure that you upload a picture of your completed notes to the document, just so that way I know that you are doing the assignments that you need to. Also, after this Edpuzzle video, there is an assignment posted on Google Classroom, so make sure you look at the agenda to see what you need to complete. So for section 4.2, and we're learning about digestion and absorption of carbohydrates. So here are your learning objectives: summarizing carbohydrate digestion and absorption, tracing the digestion and absorption of carbs, listing the enzymes used in digestion, and describing lactose intolerance. And we're going to do an activity about lactose intolerance after this Edpuzzle video. By the way, if you left your textbook at school, I scanned chapter 4, section 4.1, 4.2, and 4.3 on Google Classroom, so please use your textbook. Also, as always, the slides are posted for you. Please view them. They're there to help you. Right. So what is the ultimate goal of carbohydrate digestion? Well, like we said before, glucose is the main energy source for our bodies. And the goal of carbohydrate digestion is to break down disaccharides and polysaccharides into glucose, into smaller molecules, mostly glucose. So monosaccharides obviously don't need to be broken down at all. Disaccharides only need to be broken down once. Polysaccharides, those need extensive breakdown. How are these sugars, these disaccharides and polysaccharides, going to be broken down? Well, through hydrolysis via enzymes, right? What are enzymes? These enzymes can be found in the mouth, in your stomach, small intestine, and large intestine. So as these carbohydrates, right, move through your GI tract, starting with your mouth, chewing them, swallowing them, moving to your stomach, moving to your small intestine and your large intestine, there are different enzymes in these different parts of your GI tract that are going to be responsible for different parts of hydrolysis, right? Breaking down these molecules with water. And enzymes, their purpose is to speed this up. They're a biological catalyst. They're going to provide they're a protein. They're providing a surface for a substrate or a reactant, which would be your sugar, if it's your disaccharide, to attach to it. To speed up the process, water can come, hydrolyze it, break it down, and enzyme just speeding it up. It's not a part of the reaction. It's just a place for that substrate to bind to speed up that reaction. It's going to make it go faster. This is showing you how your carbohydrates are digested in the GI tract. So obviously, that's going to start with your mouth, right? You're going to be crushing that food with your teeth, right? So your teeth, your mouth, going to crush and tear fiber, so polysaccharides in food, and it's going to mix it with the saliva. In your saliva, you have the enzyme called amylase. And an important thing to note about enzymes end in ase, and sugars end in ose. Enzymes are specific. They are specific to a substrate. So amylase is the enzyme. What sugar do you think it is specific for? It's specific for amylose. Amylose is a starch. Amylase catalyzes the breakdown of starch. It is found in your saliva. So as you are crushing that food and it's mixing with the saliva, this enzyme is found in the saliva of your mouth, and it's going to, this is where the process of digestion is going to begin. So starch, if you're eating a starch, amylase going to catalyze that reaction. Starch is going to break down into maltose. So once that is broken down by your mouth, it's going to move to your stomach. So in your stomach, the fiber, if it's in the form of a fiber, right, it's not going to be digested. So it's going to move into the stomach. Fiber, not digested. But in your stomach, stomach acid is going to inactivate the amylase coming from your saliva. So it's going to stop and halt the starch digestion. After it moves to the stomach, it moves to the small intestine. So right here you can see that there's something called, right there, the pancreas. In the pancreas, it's producing pancreatic amylase. So you have amylase that comes from your mouth, from your saliva, and then you also have amylase in your pancreas. So the pancreas is producing pancreatic amylase, and that's going to be released into, where I put this arrow over here, the small intestine. That's going to further break down starch into, again, smaller polysaccharides or maltose. Then the small intestine is where the most digestion takes place. In the small intestine, this is where disaccharides are going to be broken down. There are 3 different enzymes in the small intestine: maltase, sucrase, lactase. Enzymes end in ase, and they are specific to the sugar that matches up with them that ends in ose. Maltose is going to be catalyzed by the enzyme maltase. Maltase, when it hydrolyzes and breaks down, it's going to turn into 2 glucose molecules. Sucrose, catalyzed by the enzyme sucrase, broken down into fructose and glucose. Lactose, catalyzed by the enzyme lactase, broken down into galactose and glucose. Now, here's something important. The intestinal cells absorb these monosaccharides. Once these disaccharides are broken up into monosaccharides, then they are going to be absorbed. Now, you also have your large intestine. So we mentioned before, right up here, that in the stomach, fiber is not digested. In the small intestine, fiber is not digested. Now, most fiber is going to pass through the digestive tract and end up here at the large intestine. Here is where some bacterial enzymes are going to break down that fiber and help some of that fiber be digested. So it's important that you see some. Some fiber catalyzed by bacterial enzymes, broken down into some fatty acids and gas. It holds water. It regulates bowel activity. It binds substances like cholesterol and other minerals and able to carry them out of the body. So fiber can help lower cholesterol. It alleviates constipation, helps regulate your bowel movements, and also, holds on to some water. So that's where the health benefits of fiber come from. Now, over here, we mentioned that in the small intestine, all these disaccharides are digested, and then they are absorbed by the intestinal cells, these monosaccharides. So I know your book briefly mentioned this. Again, I know you're not in biology, so here's just like a quick rundown. If you don't understand absorption completely, that's okay. The cells in our body have something called a cell membrane. So this is like going around the entire cell in your body. It's this green line. And what it does, it allows things to come in, also allows things to come out, right? It's not going to let something enter in your body if it's into your cells if it's harmful. So it's kind of a barrier for your cells. Going around your cell, saying, okay, you can come in or you can't. There are different ways that molecules can pass through this cell membrane. One of them being active transport, and another one being called facilitated diffusion. Active transport means that there's this protein that's going to use energy to move things into our cells. It's going to pump them into our cells. So that would be glucose and galactose. Our body needs to use energy to pump these two monosaccharides into our cells. Fructose passes through the cell membrane through facilitated diffusion. What does that mean? It doesn't need to use energy. It just flows through. So no energy used for fructose to get into our cells. Now, overall, all disaccharides are going to provide at least one molecule of glucose directly. And they can also provide the equivalent of another one indirectly. That would be through the metabolism of fructose and galactose in the liver. So the liver is going to convert fructose and galactose into glucose so our body can use it for energy. And here again, so because they're sharing a metabolic pathway, the fructose and galactose can be converted into glucose from the liver. So the next thing you're going to do is you are going to do an activity on lactose intolerance. So if you didn't, first, finish up writing your notes on this section 4.2, digestion and absorption. I know the rest of the chapter talks about lactose intolerance. So right after you finish your notes, make sure you complete that activity on Google Classroom about lactose intolerance. And it's pretty interesting. You're going to work on that for the rest of the class. And if you don't finish during our time, it'll be for your homework, okay? Also, if you didn't notice, I posted something called a discussion board, and that's going to be for any questions. So feel free, if you have any questions, to put them in there. If you go in and you see somebody has a question and you know the answer, feel free to respond with the answer. I will also be double checking to make sure that the answers that you respond with are correct. And also, if you have any feedback about the lesson today, this is very new for teachers as well as it is for you guys. So I appreciate any helpful feedback on how you think the Ed puzzle went, how you think the activities are in helping you understand this chapter. I really want to make sure that you are all getting something out of this. So please let me know how you felt today's lesson went. Okay?